We're in part 2 of chapter 6, which covers microbial nutrition and growth. We were talking about different physical restraints, characteristics of the environment that affect microbial growth. The last physical characteristic is hydrostatic pressure. This is actual physical pressure. Where it comes into play is the depths of the ocean. Water exerts pressure in proportion to its depth. Every 10 meters you go down, 1 atmosphere of pressure is increased. You can imagine how high pressure is when you are talking about going deep, deep into the ocean, like a mile down. Organisms do live there, and they're living under extreme pressure. They're called barophiles. If you remove a barophile from that extreme pressure, they will literally fall apart. Their membranes and enzymes depend on this pressure to maintain their three-dimensional shape and functional shape. If you don't keep them in the pressurized environment, the high pressure environment, they will fall apart and die. In order to study these organisms, you have to maintain a very high pressure chamber for them to thrive. Moving on from those physical traits, we return to biofilms. As you may remember, biofilms are the community of organisms that form on surfaces. In nature, this is how organisms exist. They tend not to be floating around in a liquid environment. Most microorganisms would prefer to be attached to something. Up at the top right of this slide, you see a biofilm that is formed on a contact lens. Let that visual please be a lesson to those students out there, and I know there are some of you out there that like to keep their disposable contact lenses in far longer than they are intended because you'd like to save some money. I do understand that motivation, but now that you know a bit about microbiology, I hope you realize that over time, there are biofilms forming on those contact lenses. That's why you need to dispose of them. Unchecked, those biofilms can infect your eye and lead to very serious complications and infections. You could potentially even lose your vision. It's not worth the trouble. Change out your contact lenses. Biofilms do cause other types of infections in the body. Remember, up to about a quarter of infections are really due to a biofilm that has formed somewhere within the body. The image on the bottom of the slide is providing some examples. When there is an artificial device, such as an artificial hip, implanted into the body, your normal immune system can't work as well. Biofilms could form on such a device, especially if they were not completely sterile when implanted. Those infections can be very difficult to get rid of. The same principle holds when you have something inserted, some sort of catheter or IV line. Microorganisms can grow on those inserted devices. Of course, our mouth is covered in a biofilm. Our teeth, the inside surface of our cheeks, etc., they are not sterile environments. We have many microorganisms living in there, and they're adhering to one another with their glycocalyx and attaching to the substrate, whatever that might be. Quorum sensing is a way that microorganisms communicate with each other. They actually understand what the other organisms within their population are doing because of their ability to sense chemicals that are being released by the organisms around them. If you think of a biofilm, it's really a neighborhood. Like any neighborhood, any ecosystem out there, there are different types of relationships in these biofilms. There are antagonistic relationships where different species may be competing for the same space and nutrients. There are synergistic relationships where organisms are helping each other out. One's making something that another might use. And then there's mutualistic types of relationships where they're both benefiting from the relationship. This image illustrates the idea behind quorum sensing. During quorum sensing, what is happening is that organisms are secreting a substance, and at the same time, they have a receptor for that substance so they can sense it within the environment. When there's a critical concentration of that substance, when enough microorganisms are secreting that substance into the environment, it will hit some threshold level. This is the signal the organisms are sensing. When they sense that threshold has been reached, the cells will do something. They typically turn on specific genes and off other genes. This is very important. This phenomenon was discovered with bioluminescent bacteria. Organisms would be growing in culture, and then all at one time, they all began to glow, to give off light. Researchers wondered, How is this happening? How do the cells know to all do this at the exact same time? They later discovered it had to do with this type of signaling pathway. The cells are all sensing the environment, and when that critical mass of secreting cells is reached, so that chemical signal reaches the threshold concentration, the cells turn on a gene. This happens in biofilms efficiently since the cells are held in close proximity. This biofilm structure and the ability to turn on genes can help them become more resistant to, say, antimicrobials. These quorum-sensing molecules are something that people try to study and understand better because it has implications for controlling microbial growth. This image is one that hopefully you will refer to in upcoming lab experiments. When you're sampling the environment and let microorganisms grow on a petri dish with medium, you'll notice that not all colonies look the same. The colonies come in different sizes, different shades and colors. The colonies have different types of edges, such as smooth or ruffled, and more. There's terminology that's associated with all of this. You do not have to memorize the terminology. I'm showing you this for your information and for future reference. It's very helpful to try to begin to identify a specific unknown organism by some of the characteristics. The colony characteristics will be consistent for a given species of microorganism if the environment is consistent. By consistent environment, I'm referring to keeping the culture under the same types of conditions, the same temperature, the same type of media. If a given culture is placed on the same media at the same temperature, the same type of colonies will develop. The colonies will be roughly the same size, they'll be the same color, they have the same types of edges, the colony will have the same sort of elevation, meaning when you look at the colony from the side, it will have the same overall shape to the colony. The colony characteristics give you a clue as to the type of microorganism you're dealing with. When you see a plate with many different colony types, you should realize this is a reflection of microbial diversity. Different types of colonies represent different species of organisms on that media. Let's discuss a little more about media and obtaining pure cultures. You'll have the opportunity to do a few of these key techniques in the microbiology lab. The first technique is the streak plate. The quadrant streak method is used to take an inoculum that might have mixed organisms in it and essentially dilute them across the surface of a plate by a specific pattern of spread. The goal is to enable individual cells to land far apart from each other on the media. The single cells will develop into a colony. Remember, a colony, hopefully, is a clone of one type of organism. You need one cell deposited in an area that is not close to another cell. As it begins to multiply into a visible mass, that colony will represent only one type of organism, all genetic clones. You will perform quadrant streak. Another method that might be used to obtain pure cultures from a mixed culture of cells in solution is to do dilutions before spreading. You accomplish this by taking a small volume of the solution with cells suspended in it and dilute it by mixing the volume with sterile solvent, water, or media into another tube. After you mix it up, you repeat this process and take a small volume of that mixture and move it to the next tube of diluent. The process is repeated and is termed a dilution series. Next, you would take some of those diluted solutions and spread it onto the surface of a plate. By performing a dilution before you plate some onto medium, hopefully there is now few enough cells that when you spread them onto the surface, the individual cells will land far enough apart from other cells to develop into pure colonies. When they grow into a colony, there will be only one type of organism and thus a pure culture. Dilution plating is a very common method. Pour plates are another common method, and this involves diluting your culture solution and then adding it to still liquid medium termed molten. This not yet solid medium is then literally poured into the bottom of a sterile petri dish. With this method, the bacteria are mixed with the medium and then solidify in the petri dish. Colonies will grow within the medium as well as on the surface. With the spread plate, colonies only grow on the surface of the already solidified media. There are many different types of media. We will describe them here and then use several types in lab, especially during the unknown project. For the unknown, we will use different types of media in order to obtain results that are important to identify a particular organism. Here's some terminology to become familiar with. A defined media is one where the exact concentration of every single atom is known. This may include something like calcium chloride and sodium phosphate. For these ingredients, every single atom can be accounted for in the final product. You know everything about that media. Complex media is not defined. You know what's in that medium and what components provide certain atoms, but there's mixed substances within it, so exact atomic concentrations cannot be determined. For example, a medium that contains blood or a medium that contains a digest of soy cannot be precisely defined. The media that we use in lab often, like tryptic soy agar, is a complex media because there's a component soy that's been digested up and then a specified amount of that digest is added. We know how to consistently recreate this medium, but we cannot know the very precise molecular concentration of every molecule within it. Consider a particular amino acid within this medium. We know it's contained in the medium because there is a protein, soy, and it got digested. But we don't know the exact molecular concentration of that amino acid like we would if we made a defined media with that amino acid. That describes complex media. Selective media has to have a component within it that inhibits some types of microorganisms while allowing others to grow. The selective component might be salt or a specific low pH, for example. Selective media are used to detect a subset of microbes that might be in a mixture. It inhibits some types of organisms while allowing the ones that you're interested in to grow. Differential media typically have some sort of pH indicator or something like that in the medium so that you can tell whether something does or doesn't possess a specific type of cell component, often an enzyme. This image shows a differential media. This broth that changes color. There's a pH indicator in the medium, and if the organism can ferment a sugar, acids are produced which lower the pH. The pH indicator shifts color with the change in pH, so when you see that color shift, you know that that's a positive result. We'll use several differential media tests on the unknown project. In addition, there is media that combines components so you can have a selective and differential type of media. This type of media is used when you wish to try to inhibit certain types of organisms from growing while at the same time differentiating the ones that are growing from one another. You can tell which ones are the ones you're most interested in because they could be positive for a specific color reaction. Anaerobic media has specific reducing agents within it to try to protect sensitive organisms from oxidation. Anaerobes cannot tolerate the toxic forms of oxygen. Anaerobic media with the antioxidant reducing compounds are an attempt to protect the anaerobes from being damaged. Transport media is just what it sounds like. Different types of transport media have protective substances within it to try to prevent microbes from being injured or dying on the way from the sampling site to the testing site. For example, for some of the different COVID tests, you would swab deep within your nose, and then that swab immediately went into a special tube of transport media to protect the virus in there from degrading. You wanted any virus within that sample to be detected when you ran the test. The same idea holds for other pathogens. The clinician samples the patient and then sends the sample out for a specific test. The sample is transported in protective transport media designed for that purpose. Different transport media is used for different organisms. This next image is showing a few more types of media configurations. This is a slant tube of media. The tube is filled with molten media. It is sterilized, usually within the tube, and when the liquid media comes out of the autoclave, it's cooled at a slant. Once the agar in the medium solidifies as a slant, you have a much larger surface area to grow organisms on. Here's another differential media we will be talking about in lab. This is blood agar. It's differential because if the organism can break down the blood cells, you'll be able to detect that activity. You can visually see it. On the far right is an organism that didn't do anything to the blood. The cells in the middle of the plate partially break down the blood cells. See the greenish color? Then the one on the right completely breaks down the blood cells. This next slide depicts a culture method that uses a candle jar. You probably know that if you burn a candle in a jar, it will seriously decrease the oxygen content of the jar. After burning a candle in a sealed jar, you have a very low oxygen culture condition. There are other types of specialized culture conditions. For example, you can culture animal cells under specialized conditions. That's typically referred to as mammalian cell culture. That's a more advanced technique that we don't do in microbiology lab, but there is a course in our department called cell culture where you would be trained on how to do that particular type of culturing. An enrichment culture is one where you try to encourage an organism that's in low concentration to multiply. This would be followed by another type of media to try to improve your ability to detect that microbe. A chemostat is a way to continuously culture cells. This device is something used in a lot of industrial settings. There's an illustration on the next slide I'll discuss more momentarily. In order to preserve a culture for future use, there are a few methods that may work, depending on the time frame in the future that this culture will be needed. Refrigerating cultures does okay for short periods of time, maybe a week or two. You can keep a petri plate, if you seal it, or a culture tube for a few weeks in the refrigerator before cells start to die and the culture degrades. Deep freezing allows you to store things for years. By deep freeze, I'm talking about a temperature about minus 80 degrees Celsius. You know, the original conditions required for the Pfizer vaccine. That's very cold. The best way of preserving cells is called lyophilization. This is equivalent to freeze drying. When you lyophilize a culture, you turn the cells into a powder, just like your instant coffee. When you add water to the powder, the cells come back to life. Well, this is not possible for larger organisms like ourselves. It is possible for microbes. When we purchase a culture from a scientific supplier, it typically comes in a sealed glass vial and you just add culture media to revive it because it was lyophilized before it was sent to us. This next slide illustrates a chemostat. This is a system that is used in industrial settings when they're trying to purify a product that microbes or cells of some type are producing. When you have a productive culture, it is inconvenient to keep setting up and tearing down your system because the media is old and waste has built up. For this system, what happens is that you have your culture media in the chamber and you can continuously add a little bit of fresh culture medium from the top. There is also a connection to an oxygen supply. This would have to have a filter to prevent any kind of microbe from the air from getting in there. There's a valve where you can withdraw from the system periodically to purify the desired substance. By setting up a chemostat, you don't have to keep setting up and tearing down your instrumentation, which is time-consuming and difficult. Once the chemostat is running with a productive culture, you can keep it going for a very long period of time, and you'll be able to continuously purify the product that you want. Here we're comparing different types of microbes. Microbes do not all grow at the same rate. Generation time is the concept regarding the amount of time required before a particular species will divide. When you examine these two graphs right here, you can see that the first species doesn't grow nearly as much in the 2-hour time frame as the second species. This is because the second species takes longer to go through a cell cycle of replication to divide. To give you a real-life example, E. coli can double under ideal conditions in the lab. You can get them to divide every 20 minutes. It doesn't take long to have millions or billions of cells because they'll double their population every 20 minutes, as long as they're in an ideal condition. They need enough nutrients, enough oxygen, and not too much waste buildup. On the other hand, a species like Borrelia burgdorferi, the agent of Lyme disease, the bacteria I did my thesis work on, takes 18 hours to double in the lab. At least it did back in the day when I was working with them. We had a very specific medium we had to make to get them to grow. In this specialized, highly complex medium, they would grow, but it took them 18 hours to divide. 18 hours for the population to double. So I had to wait a whole week after starting a culture in order to have enough cells to do anything. In sum, the generation time depends on the chemical and physical conditions that the organism is in, and even under ideal conditions, it will vary upon the species. This particular slide is showing you a bacterial growth curve. Understanding this graph is very important. Notice on the x-axis is time, and the y-axis is the number of cells. The cell number is increasing as you move up that y-axis. There are 4 stages to the curve displayed on this graph. The lag phase is first. This short horizontal line indicates when the cells are getting ready to divide. For example, if you move some E. coli culture to fresh media, initially the cells need to replicate their DNA, make new enzymes, and double all of the cell components within the cell. The cell itself is not dividing, but they're preparing to divide by producing all of the intracellular components needed in the daughter cells that will result after division. The log phase is when the cells are doubling. This occurs during the period of time where there's optimal conditions for the cells. This doubling period is called the log or exponential phase. During the lag phase, preparations are occurring to divide, but the cells are not dividing. During the log phase, the cells are doubling. In the stationary phase, the cell population is plateaued. The same number of cells are dividing as the ones that are dying off because they're running out of nutrients or waste products are building up. If things don't get better for that culture, then it will enter into the decline or death phase. During the decline, as the cells are dying off, replication is not occurring to replace the cells, so the number of cells decreases. This is a classic growth curve. Please understand it, be able to replicate it, and be able to talk about it. This image is depicting a specific slide that allows you to count cells directly with the microscope. There are different versions of these slides, such as a hemocytometer or a Petrov-Hauser counting chamber. We might be able to view these in the lab later in the semester. In general, these counting slides have a grid lasered onto it. You place culture and a cover slip and observe the number of cells within the grid. The volume of fluid under the cover slip is accounted for. Thus, you can count the cells within the grid and do a calculation to determine how many cells per mil are in that culture. Another direct way of measuring cells is a filtration method. This method is used when the number of cells in a volume is expected to be low. If the concentration of cells is less than about 250 per mil, you need to use a method such as filtration to detect and count those cells. This doesn't work on samples with higher numbers of cells per mil, as the filter will become crowded with trapped cells. To perform this, water samples or some other fluid are passed through a membrane that can trap cells on the surface. If we have low numbers of organisms in solution, then you can concentrate those organisms on the membrane during filtration. If, for example, there were 1 organism per mil, if you filter 100 mils, you would expect 100 cells to be trapped on the filter membrane. The membrane is then moved to the appropriate media, and the trapped cells will grow into colonies. Notice this colorized electron micrograph. You see bacteria trapped on the membrane. The little holes in the membrane allow the fluid to move through, but the bacteria are trapped on the surface. Once you have the trapped bacteria, you move the membrane to the surface of a petri dish with a specific type of media, often selective and differential. Then the colonies will change color as they grow, and you can count the ones of interest. This next method is called a viable plate count. It's measuring the cells that are capable of growing into colonies. This method is also a direct type of measurement, but it would take a lot longer to get your result than doing the count under the microscope, but it's a very common technique. To do this, you take your solution with your organisms in it and perform your dilution series. Then you take a small volume, like 100 microliters, aka 0.1 ml, from a particular dilution, and you spread plate it onto the medium of choice. After incubation, you observe the numbers of colonies that grew. Obviously, at the lower dilutions, you have much more concentrated cells. When plates, the colonies, with those plates, the colonies might be too numerous to count. As you plate more diluted cultures, the colonies are spread out. Eventually, you will have a dilution that is plated that results in colonies that can be counted accurately. Once you have the count, you can calculate your original concentration of cells. We'll do some of this in the lab. The most probable number technique, referred to as MPN, is another method that is used for solutions with low numbers of organisms. It's a method used to detect and quantify these low concentrations of microbial cells. For example, this method is used to detect E. coli in water samples. E. coli in water is an indicator of fecal contamination. You do not want to detect any in drinking water, and in water that you swim in, the numbers need to be below a certain threshold to be deemed safe. What this image is illustrating is that initially you do some dilutions of your sample, and then you use those dilutions to inoculate sets of differential media. If the organism is present and grows in the media changes color. After the incubation period, you count your positive tubes in each set, and then typically you compare your results to a chart that provides a most probable number of that detected organism in 100 milliliters of water sample. There are regulations for what's acceptable in regards to E. coli. As mentioned a moment ago, swimming water and the beach will have a low tolerance level, but drinking water cannot have any E. coli detected. The last methods described are indirect methods to detect microorganisms. One of the most common indirect methods is to use a spectrophotometer. This instrument measures how cloudy a particular solution is, so you can relate that reading to cells per mil. Spectrophotometers are very important instruments when one needs to work with specific stages of growth or specific concentrations of cells. Other indirect methods involve reactions, such as with metabolic enzymes that are able to alter a substrate into a color. How fast a color change occurs is an indication of the concentration of cells. Another way to indirectly measure cells involves centrifuging, which is spinning the cells until they form a pellet at the very bottom of the tube. You remove the liquid, dry the pellet, and weigh it. The dry weight of the pellet is an indication of how many cells you had. There are also some molecular methods to detect cells. You might, for example, try detecting DNA sequences in a sample. This is a method used especially for organisms that cannot yet be cultured in the lab. There are actually quite a lot of organisms we know exist, but we can't figure out how to get them to grow in the lab. The only way to detect them is to detect their DNA. We'll talk more about detecting DNA sequences in Chapter 8, that biotechnology chapter. So this includes the rundown on growing and quantifying organisms in lab. We've concluded Chapter 6. See you next time.